Perfect. So hi everyone. Again, welcome to another Recon community meeting. Just to give a brief reintroduction to Recon. So we are the Reagent Collaboration Network. We started during the pandemic, really focusing on connecting researchers around the world to fight COVID together, fight the pandemic, really sharing resources, sharing reagents, and promoting collaboration. And since then, we had a reboot in 2022 where we brought back our community activities, hoping to welcome researchers and scientists sharing about their work, especially with our open DNA collections that I hope many of you have heard of, maybe have used from the Open Bioeconomy Lab. And of course, we have many ongoing projects, and one of them is exactly today, our community meeting, where we are welcoming Cihan. So thank you so much for joining us today. Let me just tell a bit about you, and then I'll pass for your presentation. So Cihan is an assistant professor in the molecular biology and genetics department at the Istanbul Medeniyet University in Turkey. For his PhD, he focused on work in small molecule drug development, structural biology, and enzymology for discerning protein drug dynamics. He also has experience working with recombinant DNA technologies like CRISPR, protein expression, and purification. Nowadays, his work is on antimicrobial resistance mechanisms in bacteria, optimizing commercially important enzymes through saturation mutagenics, and working on solutions for biological degradation of PP and PE. But today, we'll hear about his personal experiences and journeys on becoming a do-it-yourself uh, worker, so someone developing do-yourself technologies, and starting a lab in Turkey under challenging economic and social circumstances. So I'll hand over to you, Cihan, and thank you again for joining us. We really look forward to hearing your lecture.
Thank you very much, Sibele. So, hello everyone. I hope everyone is hearing me nice and clear. My name is Cihan Aydin. I'm an assistant professor of molecular biology at Istanbul Medeniyet University. And first of all, I'd like to thank all of you at the Reclone community, especially Jennifer, Yanke, and Sibel for allowing me to contribute my own experiences on this subject, essentially looking outside of the box, transitioning from utilizing kits and doing DIY stuff, and essentially returning back to my own roots as a protein chemist. And essentially, today, I won't be doing too much scientific talks. I won't be talking too much about science, but I'll just give like a presentation on my development of, let me just close this down, presentation of my development as an independent scientist in Turkey, like starting from essentially the type place that I graduated after and basically roll all the way to today. So with this, let me just do it full screen. I would like to start by showing you two graphs. So the first graph that I will show you put everything that I'm going to tell you or talk to you about today in context. So this is essentially an exchange diagram for like 10 years between 2014 and 2024 between the United States dollar and the Turkish lira. So looking at this in 2014, it started somewhere around, do I have a pointer with this? Yes. So in 2015, it starts on the range around like 2.4 to 2.5 Turkish lira for a dollar. And fast forward 10 years, it's around like 32. So in the 10 years that I've been in Turkey and I was trying to establish my lab, the dollar to Turkey ratio had a devaluation over like 15-fold, like 1,500%. So it's actually a quite huge thing. And the second thing that I would like to put in perspective is the funding of the, let's say, most prominent Turkish grant called 1001 and how it scales with respect to the dollar between 2014 and 2024. So in 2013, essentially, if I remember correctly, the TÜBİTAK 1001 project will pay around like 360,000 Turkish liras. But as you can see, even with the updates on the prices, and it used to be around like $170,000. Now it basically is around like less than $60,000. So in this context, it's really hard to do something with the changing exchange ratio. And today I won't be going into too much of the details of like how things work here. So essentially, I don't know, like most of you have come from all around the world. So there are like some things which are like more specific to your country, then it's not applicable in me, but I don't know, let's continue. So I would like to talk to you first about my timeline to also put everything in context. So after graduating from college in 2007, I got accepted from UMass Medical School in Worcester, Massachusetts in America. And I did my PhD over there in like 5 years on essentially drug development against HIV-1 protease, hepatitis C, NS3-4A protease helicase complex, worked on how the protease and helicase domain of the NS3-4A communicate with each other, and successfully defended my PhD. And I was actually doing a postdoc, basically doing some wrap-up. And I really didn't have any plans to go back to Turkey at that point. I was like, okay, I'm settled down. I'm here for 5 years and everything is good. And I actually have like 2 or 3 potential jobs lined up from pharmaceutical companies back then. So life was good. Then essentially a number of familial stuff has happened, and I basically canceled all my plans, just uprooted my life that I have tried to set up in America, and basically returned back to Turkey. And while dealing with those things, like the first kind of like social upheaval in Turkey has happened. That's basically one of the largest youth movements that happened in the first millennia, called the Gezi Park protest. Maybe you know it from news, etc., or you don't. But basically, it was kind of against like the oppressive policies of the governments, and people were like just against that, and there were like huge marches and protests, and it basically went on around like for a couple of months, and I was actively participating over there. And I was actually trying to land on a small postdoc there, but since I spent more time on the protests rather than like doing my actual postdoc work, I was kindly asked to leave the lab. So that's basically kind of like a black mark on my whole career. And I spent around 8 months without anything to do, just brooding over at home, till I landed on a postdoc in 2014 September. And in the postdoc, since the PI working on there required like a person with experience in like managing a lab and essentially doing some administrative work, like work on grants, writing grants, or do like other administrative stuff, general lab management, I primarily worked on that capacity, doing a little bit of science. And essentially that was the chronobiology lab, which I started working on, and learned a bit about chronobiology, learned a bit about cell culture, and essentially formulated some projects on my head. And then I found an opportunity and started at Istanbul Medeniyet University. So that was also like a like a 6 to 9 month process where like we had like back to front talk, and since it was a governmental institution, it's also had its own procedures, so it took time. But at 2016 March, I started as an assistant professor here in this institution. And I basically had everything ready. I had like a game plan on my head. I had my projects ready, lined up, like written down. Everything was nice. And 3 or 4 months after I have started working, the failed coup attempt happened in Turkey. So it was kind of like a huge deal, like from some kind of like shadow organization in the Turkish government called FETÖ. Maybe you have heard about this. But what happened afterwards was actually quite drastic. Like anything associated with funding, including scientific fundings, were halted like around the year. So I was just ready, primed to go. And with like too many ideas, we were like, nope, we're not going to open up any grant calls for undisclosed amount of time, which passed again. But in 2018 to 2017 to 18, I got 3 grants, which was at that time worth around like $250,000. And thinking about like setting up a new lab, this money, okay, is not like too much, but it's a start. So when one USD was around like 3.6 Turkish liras, and I actually have the conversions here. I actually wrote that into my text, my grant text, where I was actually talking about the budget. So when I do the conversion, basic brutal around that. But then basically I started setting up my lab. I started working. We were essentially doing a huge number of stuff in the lab. Then the coronavirus lockdown has happened. And at that time, we need to close the lab around like 18 months. So it's a huge time for a scientist to just sit at their home, like their hands on, basically behind their bottom. So it was like a huge disappointment, and also kind of the motivation breaker at that time, because when things were going really nice and really fast, you just have to stop everything at a time, and it took like 18 months. And at that time, we were static at our homes, like cozy and nice, but the exchange ratio was basically not staying the same. Also, like the projects are like my grants lost around like 50 to 60% of their value to the dollar. So thinking about this, like why am I talking about budgets in the term of dollars? So most of the stuff that we use in the lab comes from outside sources. So they become imported. So with regards to that, anything that gets imported, you need to pay the price in dollars or in euros. So that's why your purchase power also changes with the exchange rate with regards to the funding that you get from any project. And the thing about Turkish projects is that you don't have the option to manage your own budget. So let's say you got 1 lira from a Turkish governmental institute to be utilized on a project, but the government or like the legislative function does not allow you to turn that amount to any kind of currency. So that's why it needs to stay in Turkish liras, and then with the exchange rate change, it basically lose value by every day. And when finally I finished my projects in 2022, and I finished them barely, so it was basically a race against time and race against money, I was actually in debt. And I was in debt to a couple of distributor companies. So they can basically take your word of mouth as, how can I say this? can take your words as they can trust your words and basically loan you supplies, but then basically, like a Lannister, you always need to pay your debts. And at that time, when I was into like 2022, basically I started my transition to DOI at that time, but I said that, okay, whatever stuff that I'm doing in the lab doesn't work and it's not sustainable. So I need to change what I think about. So how I think in the context of science. Maybe I need to change the subject that I'm doing. So basically then in 2003, I did kind of like a radical change and returned to my roots. So essentially I was a protein scientist before. I ventured on the drug development, cell biology level. So it was too costly. Then I basically said, okay, I'm going to close down the cell culture. I did my jobs. I finished my projects. I will then turn these into scientific papers, and I will probably venture back into protein science again, which is kind of like more manageable in my current financial situation, and which proved out to be. But talking about what I did in the lab around like, this is around like 2019, we were running 3 projects simultaneously with 4 other people in the lab, 2 masters, 1 undergrad, and a doctorate student. So we basically had a project about the generation of next generation clock reporters using nanolook or nanolook luciferase, which basically is much efficient and much strong than the regular Firefly luciferase or the Godia luciferase. We also had a project about the evaluating role of IFITAM1, which is a mammalian protein that works in immunity against viruses in clock immunity communication. And also we were developing alternate type 2 diabetes therapies, essentially through targeting biological clock components, biological clock machinery, namely cryptochrome 1 with small molecule inhibitors to see its effect on the gluconeogenetic pathways, and maybe it may improve. And we also did some animal studies to show that the compound was effective. But thinking about the methodology that we used in the lab, we did a huge number of cloning. Like we did every kind of cloning. We did restriction cloning, we did assemblies, we did golden gate, anything. And all the other processes associated with that, like picking colonies, doing mini preps, doing verification, sequencing. And then we did a number of cellular assays on that clones that we constructed. So we did cellular assays for the nanolook constructs. We did cellular assays for IFITAM knockouts, like CRISPR knockouts, or like overexpression constructs. And for the type 2 diabetes drugs, we did initial preclinical studies, also including some animal studies. So at the height of our productivity in the lab, pre-lockdown, so we used to do around like 10 to 20 clonic PCR reactions per day. So that may not be a huge number for some of you, but that was actually a huge number for us. And we actually did this daily. So we did daily PCRs, daily clonings, gel extractions, all these things. And since we are doing cloning, we need to use some kind of high fidelity enzyme, right? So the 2 things that we were using predominantly in the lab was fusion, and in like hard to do cases, Q5. So these were basically our to-go enzymes that we did. And like even with like 10 to 20 reactions, things actually start floating really fast. And we were screening around, I don't know, like around 40 colonies per day. Like at the height, I think did 200 colonies per day. At like there was like one time in the lab where they actually picked up like 6 to 8 colonies from each individual place, just grow them in tubes, mini prep them, verified with restriction. Like it was actually a huge deal, but usually it was around like 20 to 40 colonies per day. And then when we finally said that, okay, these are correct, we went on and did further essays. And most of the essays that we have done used kits, and more kits. So we had like these flash kits, Lucifer is like, we had these like, what was that called? Stop glow? No. Like most of the Promega kits about like Firefly luciferase and the nanolux luciferase. We used a number of like qPCR reagents, like involving like, and in the lab we primarily use thermoscientific, so revertate and stuff like, I don't know. Yeah, you get the meaning. And most of the time they were also, the kits were using were also black box regions. So they essentially should work like magic when you apply them correctly. But one thing that happens is that when you start spending too much money on these kind of commercial reagents, your funding starts dwindling really fast. So let's say for the dollar, when I was doing my project at the first time, a reaction of a mini prep cost around, I think, $1 to $2. Now it's €2, I know for sure, but at that time it was around $1 to $1.2 per reaction. So think about like doing 100 reactions per day. So it essentially has a cost associated, which is around like $120, which essentially is a huge bleeding wound in your budget. So essentially thinking about that, money was flowing too fast. So I didn't know where this money would go, but it was just like going so fast. And then I'm thinking about, okay, it's my first year doing my projects and I already spent half my budget. So what am I going to do about this? And this coincides with the time where like people were talking about the emergence of a virus from the Wuhan region, late 2019. And then I was like, okay, maybe we should just cut down a bit of the lab work. But thinking about grants, and you all know how grants work. You basically have these like allocations with respect to time and budgets. So you need to also finish things on time and essentially write a report about your project on time so that you can also close that and then go on to the next project. So slowing down lab work was not an option. So one thing that's preferred in Turkey is something that I call chain granting. So essentially what you do with the chain granting is that you start writing one grant after another, after another, after another. And most of the time with the second grant that you get, you finish the work that you have proposed in your first grant. And when basic the money in the second grant ends, you write the third grant so that you also finish the work in the second grant, and so on and so on and so on. Basically there are number of labs which goes like this, but I didn't find that sustainable. Why? Because like it's kind of like a never-ending spiral, like just writing grants all over the time. So where does it end? So when do you actually break even? I know a number of friends, they actually accumulate debts, they already close that gap. So in 1980s in Turkey, there were like people who were called bankers, where they would collect money from people and then they would pay people that money back with interest. But they didn't have any investments. They didn't have any kind of deals or relationship with like banks or what. They just would take money from multiple people, pay other people interest. So it's the same thing essentially. So it basically is kind of like a downward spiral. Then also looking about like things, I was also looking up like alternate revenue streams, like counseling or doing some kind of industry relationships, collaborating with industry in the context of like creating work or like doing stuff so that you can get paid on or off the books. So off the books things also we can do is kind of like the gray area, but whatever. And basically, all of these things, and it's also something that we used to joke about with a couple of friends when we were doing PhDs in United States. So let's just quit and open up a bakery so we'll earn more money. And I was actually thinking about like essentially finishing work for good. I felt like not incapable of managing a budget, and I was like, okay, kind of like a downtime in my psychological life also. Then essentially, I thought about this. So when God gives you lemons, the saying goes like this: you make a lemonade, right? No, you find a new God. So then I realized that I really need to do some kind of radical change in how I approach my work, and basically how I can save money to still produce the scientific output that I can produce, but cheaper and work smarter. So to cauterize the wound, it's actually really good to look at the cycle of essentially reduction, reuse, and recycling. So that's something that is, I think, underutilized in research. So thinking about that, I remember and statistics that I have read about, like the life sciences being one of the primary polluters with regards to plastic. So that's something that we don't usually think about. So when the money is flowing in the name of cross-contamination or in the name of like working clean, we usually don't do any kind of these approaches. Okay, so there are like sometimes that you can basically thinking about like a micropipette tip, like reusing a micropipette tip where like you're picking up like two different kinds of bacteria is a no-brainer, so you shouldn't do that. But thinking about that, like how can you save money is essentially through utilizing these three distinct parameters: reduction, the reusal, and the recycling of different kind of things. And while I was looking for alternatives to especially cut my spending on cloning, so I was bleeding too much money just on the start, just to clone stuff into, what's that? Just to clone stuff from one place to other. I didn't actually have money to do the work that is required to produce data. Then when I was doing like some soul searching on the internet, I came across, maybe you know this site, the Pipette Jockey site, Alexei. So from there, basically my total overlook has changed on how would I approach looking at the lab. So thinking about this is the first time that I have encountered the term do it yourself in the context of life sciences. So that was basically a huge life changer for me to just investigate what's going on in his webpage. And I encountered his webpage around like 2019 when I was looking for a way out. And boy, it basically produced a huge number of alternatives on how I would actually look, and essentially forced me to change my habits. So I started replacing everything that I did in my lab with cheaper and homemade alternatives. So thinking about that, I had like a couple of vials of fusion lying around, and I used to do 50 microliter experiments. Now today I'm doing actually like 10 microliter PCR experiments, but whatever, that's another case. So instead of using fusion, essentially fusion is something that most of us who are invested in cloning utilize in the lab, right? But homemade fusion is a better alternative in this context. And another, like for my case, another huge money output was for the DNA RNA prep columns. I tried a number of different replacement ways to cut money on the DNA prep columns. So I did like regular alkaline lysis with ethanol precipitation. I tried diatomous earth. I tried the silica gel method. None of them actually worked as clean as mini preps, so that's why I always was using mini preps, but then I learned that you can actually recycle them and reuse them with homemade reagents. So RNA extraction, we also used to do a lot of like RNA extraction work for qPCR, and that was also kind of like a money bleeder. So then we actually turned to homemade triazole, which I think a guy in plant sciences was actually putting on his page. So nowadays, restriction enzymes, it's still something that we use a lot in the lab, but now I am actually transitioning to homemade restriction enzymes. So that was kind of like a learning curve in the process, which I'll talk about. So qPCR, I start using homemade RT. So also homemade murine RNase inhibitor, uridine DNA glycosylase. Plus, I also started buying my cyber green bulk from China so that I would actually hello? Okay. So I was actually started buying my cyber green from bulk from China so that I would actually per reaction would essentially make it more economical. So I would only pay 10% of what I used to pay per reaction to synthesize and then analyze the expression of different gene products. For luminescence, like we did a number of bioluminescence and luminescence-based endpoint assays with Promega kits. I actually turned totally homemade with homemade luciferin, covalent resin. Even we utilized covalent resin H in our like nanoloop constructs, which didn't work as nice as the Promega, but still it worked. So, and a huge number of other things that actually allowed me to save money on the lab. So these kind of approaches are actually not that hard to take. So that's what I discovered when I was doing that. I was actually fearful at first about the cross-contamination. I feared about like not having compatible results. But that's the thing, like it's just a learning curve. Like with the learning curve, you actually adjust on your current situation, develop your methodologies, optimize, and then essentially go into production mode. And that's basically what I did. So giving like a small example in the context of the reusing of mini prep columns, there are actually also published articles where people also took into this issue and developed strategies to clean the columns so that you will have columns that are cleaned and essentially that have negligible cross-contamination from carryover DNA from earlier prep results, which basically allows you and allowed me to reuse my columns over and over again. So with regards to that, if we talk about the small protocol on plasmid regeneration, basically you can do a water wash just to get rid of extra salts or whatever. Then you can wash it with 1 molar hydrochloric acid or phosphoric acid. My preference is hydrochloric. You can do it 4 times. You can leave it overnight. You can leave it for a month. Nothing happens. Then again, after the acid wash, you do a water wash and a 70% ethanol wash, and you can just try it. And you with this, you basically have negligible carryover contamination. So what those articles have, like those articles provide, not provide, but those articles suggest that they don't have carryover contamination, and they actually elucidated their point with doing qPCR on like columns that are prepared that basically didn't like show like high CT values, like really high CT values. And then you can, after drying, you can reuse it your hard contents. So in this context, I used and reused my columns like more than 10 times and still did comparable results. So think about this. You can just take a single column. It doesn't matter from which kit. it may be Kiagen, MN, or nowadays what I do is I actually buy bulk columns from China. And I have like 2 vendors that I really trust. I can just give the names. If you want, you can just send me an email or whatever. I can just let them contact you. And basically even also regenerate them. So to lower my cost even less. Just thinking about that, you can reuse your columns to make clean DNA or clean RNA, and it works essentially. So if it works, why not do it? And then you essentially can do your mini prep with leftover reagents, or you can prepare your own reagents. So any kind of like silica column that you use in the lab is compatible with this procedure. So you can prepare the reagents with regards to the Kiagen formula. So with resuspension, you can have something like this. And note that I actually keep the RNase A concentration quite low. So the recommended is 100 to 200. I saw that 20 is more than enough. So this is an extra money saving step. So with the alkaline lysis, you have the sodium hydroxide SDS. And for neutralization, the only thing that actually costs here is the guanidinium hydrochloride. So you can buy it in like 5 or 10 kilograms. So I actually bought a drum of guanidinium hydrochloride from China, which I'm using. And it basically was 5 times less the price that I got from Acros. And then basically you have the first wash. If you are using NDA plus strains like SDBL3, then you do your regular wash. And essentially you do the prep as always using the Kiagen methodology. And it actually works. And even if like after 10 or 15 times cleaning my columns, and not in a trial way, but in an actual experimental setting, I didn't see any cross-contamination. That I did sequencing on them, I did functional assays on them, I did cloning with them. No, like it works. So then what happens is that, thinking about like starting with something like MM column macronasal, you start from $2 on the reaction, then you can go down to 0.1. So with the Chinese columns that I use, my cost is less than 1 cent per reaction, since I'm also regenerating those columns. So that's a huge money save. So that makes cloning like so much easier in this context. Why? Because like you can just do mini preps to your heart's content without just thinking about the consequences. So that's basically what I mean. Another thing that we can do is, and it's actually quite easy to do, is to purify and utilize, and even like not purify, but use your own fusion polymerase. So there are the number of people who has the fusion plasmid, or what you call the PFU SSO7D plasmid available. So I think the Free Genes had one clone. There's a clone roaming around the world from the Barrick lab. So basically what you can do is you can transform it in BL21 D3 cells, and then you express them. And what you do is that you just do a quick heat lysis step. So 70 degrees, 1 hour is more than enough to heat lyse E. coli, denature most of the proteins, so that only what you have is your fusion polymerase and some negligible contaminants. And what I do at this step is also, after the heat lysis, I precipitate extra DNA with streptomycin. In some cases, you can also use PEI, but streptomycin is my preference because I can essentially go to a doctor, just beg him to prescribe me with streptomycin, and we'll get these injectable streptomycin vials that we can create streptomycin from. So it basically is 100 times cheaper than buying it from Sigma. Yes, it's actually 100 times cheaper. I did the math. And then you aliquot and store in freezer in -20, and then you can use it whenever you want. So one thing that I would actually recommend at this point with regards to the fusion polymerase is to buy the buffer. So the buffer actually comes cheaper than if you're actually preparing the buffer yourself. So as you can see, the real buffer itself actually is composed of many things, which is kind of like handful and hard to find. But the original buffer actually comes around, like, if I remember correctly, $8 per vial. So $8, 1.5 mLs of 5x buffer will be enough for like a huge number of reactions. I couldn't do the math. Okay. But then it all becomes like this. So can you really go self-sufficient? In the context of molecular biology, of course. So there are like some things that you actually need to buy, and that also involves equipment. But one thing about like going really self-sufficient, especially on the core part of the molecular biology, is the ability to construct your own parts and essentially create our own solutions, custom solutions. And that is basically where I started doing my actual transition from the cellular biology aspect and thinking really hard about like reading and investigating more about like synthetic biology, and essentially like returning to my roots about like protein engineering and whatever, so go on. And essentially the open bioeconomy genes involving the open enzymes, which has a number of different things that is really paramount to have in a molecular biology lab, that will allow you to do anything actually, essentially. The open reporters involving chromogenic reporters and fluorescent reporters, the open plasmids that you can construct your assemblies on those, and also the molecular diagnostic kits. Okay, you can use it. It's pretty much ready to go enzymes that you use for molecular diagnostic, and the Bacillus subtilis protein secretion toolkit, which I actually lack. I actually, if I could get my hand off these plasmids, that would be really good. So if like some of you like have it, I will be really happy if you can share it with me. That will allow you to essentially check a number of different secretion tags in the context of the Bacillus secretion to see which tag works more efficiently with your protein system. And if there's something that doesn't be in here, so thinking about that, you also, in the Stanford genes, you have the organismal gene sets, like E. coli protein expression set, you have the open yeast collection. So a huge number of different stuff that will actually help you to construct anything that you need in the lab, actually. And if there is something that is lacking, you can integrate as new parts. So that's the beauty of the molecular system, essentially. So you can create your own parts, and then you can integrate to the system. So with regards to that, I actually started transitioning with regards to some of the restriction enzymes from what I usually use, which I use mostly thermo fast digest enzymes, but I transitioned especially for like let's say EcoR5, Eco31I, which is a type 2S enzyme, BSA1, from buying them commercially to actually produce them in lab. So what we do with regards to our restriction enzymes is that you can clone the restriction enzyme in a plasmid. Like you can assemble it with regards to different components that you get with the E. coli protein expression toolkit, assemble them into a complete plasmid with the appropriate promoter and appropriate backbone. So in my case, I usually have a standard system for the assembly of the restriction enzyme plasmid itself. I use the HISTAC version with the T7-LEC-O promoter and the 0034 RBS. I use a custom PET style backbone, so it has like a couple of custom modifications that I have, and I actually integrated into the open enzyme system in an AF part format. And then you also need to prepare and plasmid for the metal transferase. So each restriction enzyme also comes with its own metal transferase. So if you look at the list in the open enzyme list, you can actually see that there's the metal transferase associated with that to a backbone that I generated from P15A, which essentially houses the chloramphenicol resistance. So I can have essentially kanamycin for the restriction enzyme, and then I can have the chloramphenicol for the metal transferase. But this time I use a promoter. Maybe some of you may know this, the OSM-Y promoter, which is a stationary phase promoter that I use, so that I get kind of like a tiny expression, but when I hit stationary phase, it basically becomes maximally expressed. Then I hit it with the IPTG, or in some cases, I use autoinduction media, so that I do my expression. And then I purify the enzyme using nickel NTA. So I actually use a cheaper version of the nickel NTA based on IDA that I buy from China. So I also cut money from there instead of getting like my column material from Kiagen. I just get it from some kind of generic source in China. Not as efficient, but it does the job. So that's basically the important part at this point. And then new part integration. So anything that you need as an extra part, and if provided that you're going to use it constantly. So I have like an array of solubility tags that I use. I use spin-drawing solubility tag. I use the trigger factor, which were not included in the original open enzymes toolbox, but I don't know if they are not involved because they are commercial, they are new, or they are patented. I haven't checked that, but one thing that you can do is that you can PCR your new part with appropriate oligos. So also thinking about the overhangs that you require. So if you're like creating an N2, N3 part, you need to have the specific overhangs suitable to create an N2, N3 part so that you have the correct amplicon. And I actually ligate them into my own spin on the PJET. So if you have like, I don't know if there are like any of you who previously used the PJET system to do cloning or subcloning, but it's advantageous because it expresses a restriction enzyme when it's ligated, and the restriction enzyme will kill the bacteria. But if you integrate something inside it, it basically disrupts the coding sequence for the ECO 47I restriction enzyme. So you basically don't have like a dead cell versus living cell background instead of like blue-white screening, which also costs money due to IPTG. Sorry, not IPTG, but XGAL. And then you can use these plasmids directly in the Golden Gate assembly. So what I do is I actually inserted a small fragment, ligated a small fragment in the original linearized vector coming from the CloneJet kit, like I got back in, I don't know, like 2016. And I basically, what I do is I just cut this with Echo R5 or Echo 32I when you come from the thermoscientific side or fermenter side, and then you get the linearized vector. But otherwise, the vector itself can propagate because it basically has its own gene function disrupted due to this insertion, which basically puts everything after this insertion out of frame. So that's something that I do, and I basically utilize this for like the last 7 years now, which worked wonders in any kind of like situation where you need like a subcloning or an intermediate cloning vector. And then you can use whatever that you have created as a part, integrated into this PJET variant in anywhere in your Golden Gate assembly. And I also have like 3 different variants with the chloramphenicol resistance, ampicillin resistance, and the tetracycline resistant markers, so that I can actually switch between vectors with regards to the level of assemblies that I do, so that I don't have any background contamination when doing assembly. So that's also a plus. So thinking about that, the possibilities that you can have with all of these do-it-by yourself reagents, like about like the information snippets that you get from forums, the information snippets that you get from blogs, and like basically with talking to other people in communities about like open bioeconomy, like here in Reclone, the possibilities are essentially limitless if you're motivated enough. And the motivation that can come from necessities, or you may be actually motivated yourself, is really important to drive this pathway forward. And I think I'll just finish my presentation about doing a pros and cons. So thinking about the pros, and I was actually going to add two more things here. The first pro that comes into mind is sustainability. So when you like use the recycle, reuse, and I forgot the third one. The 3 R's approach, you basically form a sustainable environment. So by reusing, you can also save money, but you can also save resources. So that, and also with the addition of the systems that allow you to mix and match and create your own genes, will allow you to become a more sustainable. So I'm actually now, I got a project last year, which basically came around like $50,000 to $60,000, right? Yeah, $50,000 to $60,000, and by just following these approaches, I still have like $30,000. I don't know where to spend. Like I bought everything for the project, and everything is ready. And I actually did some stock because we are going to have an election in the end of March. And basically, after every election, the Turkish lira to dollar ratio jumps up. So that's basically why I did stock. But I still have money to spend, and I basically like trying to spend it before the election. But still, anything that you do while transitioning to DIY essentially allows you to form a sustainable and economical lab. And one other thing is that especially for countries which require distributors. So let's say, I don't know, but in Turkey, what happens is that basically the companies that you buy stuff from have these local distributors, and whenever you want to buy something, they won't have it in stock, and you need to wait for like 2 months so that it will come from like a shipment and go through the customs, enter the Turkey, like all these procedures and whatnot. You need to wait for 2 months. That also makes you self-reliant. So you can essentially create your own enzymes, purify them, and you can arrange your time in a way that basically nothing will be disrupted due to a lack of a resource. And also these kind of systems allows you to be flexible. So I have a recent example about that concerning the open yeast collection system. Yes, open yeast collection. So essentially, we were looking for a way to overexpress and do some CRISPR-based knockout on a yeast system. And I was actually looking at plasmids from Edgene, and I said, okay, I need to buy this, buy this, buy this. Then I just remembered I have the open yeast system. Okay, now I can just buy 3 sets of primers and just essentially PCR my gene of interest, integrate them to the open yeast collection. No, not integrated to open yeast collection, but integrate them as an entry vector to the open yeast collection, like make it suitable and compatible, and you can just like mix and match. So that's basically something that was not previously achievable by old style thinking. And also with this, you know about know-how of like how things work. So, and when you do invest time in DIY, you first need to learn what does what. And by learning this, you actually increase your knowledge level so that you actually know, instead of like just approaching to some things as the black box approach or like a black magic voodoo, so that say, okay, this works. You don't know why it's working, but it works. You actually know why it's working. And with all of these actually pave the way to availability of pursuing different paths. So thinking about the foundation behind the open bioeconomy approach is to make people, like avail people to, can I say this? Avail people to also think in different ways. So let's say you have some kind of product that you produce through utilization of the anything associated with open bioeconomy systems, and then you're basically thinking, okay, maybe it's time to commercialize this. Like all the ways are open for you. Otherwise, you would just get stuck behind like licenses, you'd get stuck behind like patents and all these different kind of things. But that's also a plus approach when you are thinking on more like a next level kind of thing. And thinking about the cons, on the other hand, most of the time, since you're just basically transitioning your mindset from kind of like a closed, ready-to-use environment to some kind of open environment, you also need to invest more time. So it's not like as just a get the kit, do the reaction, done. It's like you need to prepare, you need to be ready, you need to optimize. Everything about this actually takes time. And since there are a huge number of moving parts in these kind of like DIY systems, it's also makes troubleshooting much harder. So that's something I struggle constantly when I basically am creating new systems, testing them. So thinking about this, okay, like I used 5 new stuff here, so I need to just check them one by one to see what does and what doesn't work. And it also involves a huge learning curve. So basically transition from closed source to open source, you need to learn about what goes in what. It's actually a good experience in my case, like in when I think about it, just to learn about stuff. I like it, but still for some people, especially who are not very well versed in protein science, it may be a bit of a steep learning curve. So, but once you overcome, I think the pros way outweigh the cons. And with this, I would like to thank you all for participating in my presentation about my personal experience in transition DIY. So if you would like to contact me from social media, from LinkedIn, Instagram as Twitter, and kind of like not scientific, more personal, but still, in any cases, you can just use my handle, Cihan Aydin PhD. Or if you'd like to contact me further about like anything that you'd like to ask about, let me just do this really quickly. You can email me using my email, which I am writing down here. cihan.aydin@gmail.com. So with this, thank you very much, and I hope you enjoyed my talk.
Awesome. Thank you. Thank you so much for sharing your journey. We had also quite some conversation happening on the chat with everything that you were presenting. It was quite nice because, any questions we had, you kind of answer telling your story, which it's amazing. And a lot of people talking about the different open DNA collections, some work coming up that I know you're on the list to receive from Recon. Some of the kits we're working on, and we had many of other people interested in that.
Oh,
And
I'm reading.
Yeah,
Oh.
awesome. Yeah, perfect. And we are on the time for the session, so if anyone needs to leave, that's totally okay. Yankee sent on the chat the link to our forum, so we can definitely continue the conversation there. But also, if anyone has questions, and if Jihan doesn't need to leave so soon, we could
I'm
definitely
not pressed
open.
for time.
Perfect. Then we have time for 1 or 2 questions. If anyone wants to raise their hand or type on the chat.
I mean, I did have a quick question earlier on, but I think you sort of answered it in your pros and cons list because I was going to ask about the balance between the reduction of cost, but also the time needed and kind of know how to kind of learn how to make all these homebrew kits.
So, sorry.
No.
the thing about like pros and cons is that like I usually weigh about like how many times I'm going to use something. Let's say like for the regeneration and essentially doing the mini prep columns, I'm doing like at least 40 to 60 mini preps a week, even today. Like even today I do it like that. And thinking about it, it's that it makes sense as an investment, but let's say thinking about like something that I'm going to do once in my lifetime, then it's kind of like a think about it. So do I need to invest this much time and this much of myself? So will it justify it? So if the money on the other hand is too steep, then again the answer is yes. But if the money is manageable, so think about like the fusion buffer. So the actual fusion buffer has too many moving components. And then thinking about that, so I just buy it. It's just not worth my time for $8, but for something that I can do it for like, let's say at the cost of $100, where I need to buy it like in the console like $3,000-4,000, where there's a huge price discrepancy, I'll just bite the bullet and just do it.
And I believe, Adrian, would you like to ask your question or should I read it out for you?
OS best solutions.
Open source.
Open source best solutions.
So all these sources, the solutions that you, so I'm running a DIY club in Ottawa here, and yeah, so sometimes we buy the kit, but it's expensive and it's hard for the members to pull out the numbers for that. I was wondering if you have a place where you kind of documented some of these protocols and maybe the vendors and stuff like that, if we can really use that approach.
So one thing about this is I usually do everything from memory, but I can definitely write down the protocols, like write down the common protocols that I use in the lab for doing like some of the stuff here. And also I can provide you with a vendor list. So most of the vendors that I buy my stuff from, especially in the context of chemicals and plastics, they are not like the big names nowadays. Like they all come from China. And again, like I basically collaborate with a distributor who goes and comes back to China so that I don't need to deal with customs, because customs is a huge deal in Turkey and you actually need to know how it works in order to get your stuff through. But I have like a couple of firms where I actually like the products, and some of the people that like some of the companies that I buy chemicals from actually are producers for Sigma. So they are actually they have pretty much good chemicals. So one example is this company, BLD. So I have written it to the chat, which basically produces high quality chemicals at I think 10th of the price. So Sigma just sticks up the Sigma and basically the price bumps up like 10 times. But I can definitely create kind of like a list. If that's something that everyone wants, I can just like either write like a forum post or compile something and put it somewhere so that it can be accessible by everyone.
That's definitely something that I think many of the members in the Recon community would second, having some documentation about these open source protocols. And I believe we shared with you, Cihan, the Recon Protocols IO page as well. So we can certainly help you with making sure that those are open and available.
I stalked through the forum.
Yes, I think a lot of us do. Sorry,
maybe you can become the next pipette jockey there. Because Alex actually, I'm not sure if you know Alex.
I actually know Alex. We had a number of email correspondences back and forth.
Yeah, he's from Canada, so we used to cooperate with something. But he actually he finished with the academia and he works for a company.
I know.
And he's doing a self-free system, so he cannot continue to publish those great articles that he got you started on this.
No, that's the thing though, like actually like talking with him and reading through his blog basically initiated me to think this way. So I have used his phase separation gel. I actually did a lot of back and forward when we were doing this like mini prep cleanups for DNA and RNA columns. So we did kind of like our own spin on RNA. And yeah, that's basically it. Like I think he went to dark side. I think he went to dark side.
He had no choice.
Yeah, but I think they have a really good system based on like some kind of membrane-based system for cell-free protein expression, and it's never really he's doing really interesting stuff.
Yeah, and it's not extremely expensive either compared to all the other ones.
Oh yeah, oh yeah. Okay, any questions?
Then I think that's all the questions I had for the moment. But that's definitely feel free to continue the conversation in our forum. Thank you so much for accepting our invitation for today and sharing about your work.
So again, if you'd like to contact me, just shoot me an email. And I need to say this first, I am bad with email, but I'll probably like return after at most a week. But yeah, like if you have pressing issues, questions, or like any kind of like collaboration that you want to set, I'm open to all like I'd like to talk about these stuff. So you can just shoot me an email. Okay.
Thank you. Yes,
Thank
and
you
thank you
again.
everyone for joining us today.
And thank you everyone for listening to me, and thank you all for like just inviting me to give this seminar. Okay, take care guys. Bye bye.